Delighted to have Dr. Segun Fatuma speak to us today. So just to give you a little bit of background, Dr. Segun Fatuma is a computational geneticist and Associate Professor of Genetic Epidemiology and Bioinformatics at the London School for Hygiene and Tropical Medicine. He's also a Fellow of the Advanced HE and, as of July 2020, a Wellcome International Intermediate Fellow. So very topically, he leads the African Computational Genomics Research Group at the Medical Research Council, Uganda Virus Research Institute, and London School of Hygiene and Tropical Medicine, Uganda Research Unit. That's such a big mouthful. So for short, that's just the MRC UVRI and the London School. So Dr. Fatuma's research largely focuses on the genetic impact on non-communicable diseases in Africa. And he has a particular interest in assessing the impact of genetic variation on kidney function, diabetes, lipid metabolism, and a whole range of cardiovascular and cardiometabolic traits. And he and his group use very different approaches. So they use genome-wide association studies. They look at polygenic risk scores. They do fine mapping, for which African genomes are ideal. And they also do studies on Mendelian randomization to look at causality, as well as thinking about discovering new drug targets. So very much of interest, and in the context of his presentation today, Dr. Fatuma co-leads the Nigerian Non-Communicable Diseases Genetic Heritage Study and a consortium, and co-directs the Nigerian Cardiometabolic Genome Study. So he's very actively involved in building capacity across Africa and works in Uganda, Mali, Nigeria, amongst many others. So Segun, just to say, I think you are becoming so well known to this community now for the incredible work that you're doing, and we welcome you to talk to us today about cohorts and what you think is happening in Africa, success stories, and much more. So over to you, and thank you so much.
Thank you very, very much, Michelle. I feel very, very privileged to be here today. Thank you very much for the introduction. I'm going to share my slide and make my presentation. Okay, so good afternoon, everybody. Today, I'll be speaking on a presentation that I've titled How to Navigate the Global Biobank Bias for Genomic Study, and what can we learn from flourishing African biobank. So I always like to start with this slide, and the reason is because I don't like to make an assumption about whether people understand Africa and what goes in Africa. So Africa currently has a population of about 1.4 billion people. That is 1.4 billion people. That's a lot of people. So Africa is the world's second largest and second most populous continent in the world. And the reason why I talk about this is when I go for meetings or somewhere, and I tell people that I'm from Nigeria, several occasions people have told me that I have a friend in Zambia. Do you know him? Of course, I don't know him, because there are 1.4 billion people in Africa, and there are 54 countries, not a village. So Africa has more than 3,000 different ethnic groups, including about 2,100 different languages. So currently, about 50% of African population are children under 18 years old. For example, in Uganda, the average age in Uganda is about 16.7 years old. So if you compare that to the UK, which has an average age of 40 or more, you see that the population in Africa is very, very young. And there's implication for this for genomic study or medical research generally. So by 2030, African under 18 population will increase by nearly 170 million people. And in the next few years, by 2100, 50% of the world children under 18 will be living in Africa. So these are very, very important facts to know. So why is African genome so important? There have been several studies that have confirmed the theory that is called the Art of Africa theory. So this theory is about the modern woman who's evolved from Africa and then migrated through Europe and Asia and reached out to the Pacific and as well as America. So as we know, African population today have more genetic diversity than any other population in the world. And this genetic diversity found in non-African represents only a subset. So what you find in the European population or in Asian population, other population, they are more or less like a subset of what you see in African population. So here you will see the map of Africa here where I'm highlighting, showing so much of diversity. And you see from other population, which is more or less like a subset of what you have in the African population. So even in African population, so larger genetic differences happen within African population, within different regions of African population. For example, within Western region or Eastern region of South Africa. So there's much more genetic diversity within Africa, even than between Africa and European population. So, and all these have implications. So first, genetic study of African population are crucial to fully understand the modern human evolution and the effect that it has on human disease. Secondly, so the finding implies that the result from genetic study in non-African population, including their clinical relevance, they may not be generalizable to African populations. So we must understand that there's much more diversity in the African population, which must be well captured. So thirdly, so this implies that there are many genetic variants yet to be discovered, because what we found in the European population, Asian population are more or less like a subset of what you have in African population. So there are still many genetic loci that need to be found in African population. So this finding implies that the result from genetic study in one subpopulation within Africa may not also apply to other populations. Like I said, there's so much more diversity within African population, and the result that we found from, say, West African population may not be transferable to East African population or South African population. So unfortunately, there's a bias in the current global biobank, which are used for genomic study. So according to one research study that we did, we published this year in Nature Medicine, so we showed that 86%, that's more or less like about nearly 9 of every genomic study that have been conducted so far, they were done in individual of European ancestry. This is happening despite the scientific rationale for including African population. Only 1.1% of African participants are currently in GWAS catalog. So here on this slide, so I show a diagram showing a well-coordinated global effort, which is aiming to aggregate global biobank for genomic study, which include about 2.2 million people across the world. So what we see is that you see biobank all across the world, and if you look here at the middle here, Africa population, you find none. So here again, so showing that scientists tends to utilize data sets that were created and are easy to access. But this is now changing because we are now part of this global initiative. So here I show a paper that was published just a few weeks ago in last month. This paper is a saturated map of common genetic variants. So more or less, what this paper is saying that we found everything that has to do with loci that are associated with height. So in this work, they put together a study that include 5.4 million people, including more than 280 GWAS. They were aggregated globally. None of this study, none of those 281 is from continental Africa. Of course, the paper showed that there's one from Nigeria. That one from Nigeria was actually part of a study from the UK. So invariably, there's no study from African, continental Africa in this paper. This particular paper, for example, has more than 600 authors, and they claim to find all association. So what I want to bring out here is that it's not that there are no GWAS of highs in continental Africa. There are, of course, there are some in Uganda, in South Africa, from Haugen, and other places. But you see again here that scientists tend to exclude study from continental Africa. So the question is that why exclude continental Africa as study from this kind of population? That might not be right. So here I show a diagram from one of my colleagues, Caroline. This is now UK Biobank. So UK Biobank is one of the well-characterized cohorts in the world that are being used globally. It has about 500,000 participants from the UK. And those participants include about 30,000 individuals who are non-European ancestry. So in this plot that was done recently, we saw that 93% of publication excluded non-European sample in their paper, in their analysis. So what we see here is that scientists like a lot to talk about the need for diversity, but when they have the opportunity to include or to analyze data from continental Africa, they exclude it. So scientists need to walk the talk. Yeah, so here I show how we are progressing in the last few years. And on the left here, you see the representation of different population in GWAS catalog. In 2016, for example, we have 81% representing European population and 3% in Africa. In 2019, another paper, you see that the African population has now decreased from 3% to 2.03% in 2019. 3 years down the line in 2022, African population is now 1.1%. We are not doing very well. You see that the population of European population is increasing and African population is decreasing. Bearing in mind that there is more diversity in African population, there are so many variants that need to be discovered that stay in African population. So despite the repeated warning and call, genomic eurocentric bias is on the rise, and we need to find a way to fix that. So what I would like to say here is that even the 1.1% Africans in genomic study, they are people of African Americans. And here I want to make a call, which I think is very important. So the call is that we commend the excellent efforts to improve representation of African ancestral individuals in genomic study. And you see there are so many studies, such as a PAGE study, Emerge Network, there's MVP, TopMed, trying to improve representation. That is well commended. But the truth is that the growing genomic data from African Americans and other African in diaspora don't and can't tell the full story about the value of African genomics, full stop. So we need to be very clear that trying to just keep on aggregating data from African Americans and African in diaspora would not tell the whole story. And we have to put this in mind. So again, just to emphasize what I said currently, is that if we continue to study a small number of African diaspora population, including African American, Black participants in the UK and Europe, and grouping all of them together into a broad category of African ancestry, we will continue to promote genomic imbalance and widen ethnic disparity, and we will fail to capture the genetic diversity in Africa. So I'd like to emphasize again that in Africa, there are more than 3,000 ethnic groups. Many of them are not captured. So what we see in African American and Black participants are just a very tiny proportion of Africans. So we must think about how we will capture the genetic diversity in African population, and I mean continental African population. So in order to improve representation of African in genomic study, we must make effort to engage, to recruit participants and analyze data from indigenous African population. And if you ask from people that are leading on the UK Biobank, and other great projects abroad, what you will hear in terms of why there is low representation is that the population of Africa are always hard to reach. But that's not the same thing. We don't have the same experience in African population. I'll now be sharing our experience, what we have. So I'm currently leading the Uganda General Resource in Uganda, and this cohort has a population of about 22,000 people. We have genotype 5,000 participants on 2.5 million Illumina array. We have additional 2,000 that have been whole genome sequenced. We have a plan to expand this study to include proteomics, metabolomics, epigenomics, and single cell genomics. So most of the talk will be focusing on what we have done so far on the Ugandan Genome Resource. And currently also, we are now generating what we call the Malawi Genome Resource. So Malawi Genome Resource is coming out of the Meru cohort that is led by Professor Mia Crampin in Malawi. So that cohort has about 25,000 people. So there are samples that have been collected from rural and urban community in Malawi. So currently we're genotyping 7,000 individuals on East Africa array, and there's a plan for expansion. And so towards the end, I will talk briefly about the NCDGS consortium, which aims to collect 100,000 samples in Nigeria. So this consortium is co-led by many people, about 5 people, including myself. So this is a private-public partnership looking at multi-center across Nigeria, including projects in the community and as well as in the hospital. So the plan is to do whole genome sequencing, whole exome sequencing, and genotyping. And it's a plan to expand this also across Africa. So I'm going to start with the Uganda project. So we have the rural community in Uganda called Shamulibwa. So Shamulibwa has about 15 villages, 22,000 people, and here just showing a map of Shamulibwa. So this cohort has been in existence since 1989. So from 1989 to 2012, the focus had primarily been on HIV surveillance, trying to understand the prevalence of HIV in Uganda population. So since 1989, we've done like an annual survey from door to door, interviewing the participants, and also do like a manual data entry. But in around 2011, 2012, we've now focusing a bit more on non-communicable disease, including doing genomic studies. So what the study I'll be describing today will be the genomic study that we did from this cohort from 2011. So even currently, from 2017 to 2022, we still continue our genomic study, and also including other studies, including COVID-19, cancer study, and others. So 2019, so we published a paper that was described as the largest ever genomic study of Africa. So don't forget that I showed previously a paper from the Global Consortium that described 5.4 million people. But what was described here as the largest ever genomic study of Africa in 2019 has only 5,000 people that were genotyped and 2,000 whole genome sequencing, just to see the disparity. So here is describing about 2,000 participants from Uganda, and you can see a lot of diversity from just about 10 different ethnic groups in a local community in Uganda. So you see evidence of population substructure, potential confounding, and this will need to apply a different model or a mixed model for this kind of genome-wide association study. So as you may expect, here we found a higher level of genetic diversity in Uganda population than what you see in similar study of European population. So around a quarter of genetic variation identified in this Uganda population had not been discovered before. So specifically, we found that 28.7% of SNP discovered in Uganda whole genome sequence dataset were not found in genome aggregation databases. So specifically, this 9.5 million novel variants that are not represented in either 1,000 genome project phase 3, African Genome Resource project, and as well as the UK 10K reference panel. So this shows that a lot that we're missing if we continue to exclude African from genomic study. So we demonstrate the value of African Genome Resource by creating African unique reference panel using data set from the 2,000 whole genome sequence that I mentioned previously, and also data from the African Genome Resource project and 1,000 genome projects. So as you can see here on this figure, on the top left, for example, so our unique reference panel has performed existing imputation reference panel, particularly when you do imputation on East and South African population. So here, this table is showing the results that we have from GWAS study, GWAS, on different traits, different non-communicable traits. So you can see that we found some novel association. Just bear in mind that it's just a sample size of about 5,000, 2,000, and addition of a few others from African population. What I really want to emphasize on this slide is the last two columns here. So the second column here, which is a minor allele frequency for African population, and the last one here is the minor allele frequency of the European population. So what you will see in the minor allele frequency of the European population is that you will see that this is mainly a 0 across board. So meaning that these variants are monomorphic in European population, or they are absent in European population, which explain the reason why we find them, because they are not present in European population. So our study also leveraged on the lower linkage disequilibrium in African data sets. So our fine mapping analysis resulted in a narrow credible interval at most traits. So we're able to map 16 out of the 41 distinct loci to a single SNP in the credible set. We're also able to resolve the previously identified association with the HB1C. So, very interesting here, we assess the heritability of all the 34 cardiometabolic traits in this study. So we found that height, for example, is highly genetically determined in Uganda if you compare that to what you will see in European population. So the narrow sense heritability of height was 49% in Uganda compared to about 90%, 70% to 8% in European population. And in contrast to that, the heritability estimate of LDL were significantly higher in Uganda to European population. So you see different heritability for different traits. So collectively, what it shows us is that our findings from a small data set has a great potential and value. It has great potential and value of showed a great potential and value of African genome data for discovery. So most importantly that I want to see today is to show you an example of our community support for research participants that we use that is aiming to build cohesion and to build support to show how we interact with our research participants in the community. So this is just an example of a recent interaction that we had with the participant in January of this year. So we show up and there are so many activities, so many sports, and I will show you the reason why this is very important for this particular community. This is one example of such. And it's just an example also showing, providing support for the community, engaging the community, and making the community feel supported even while a study is going on. And here's also another example of how we interact with the participants. And here's another example, and another example, and many examples. So what can we learn from the Uganda cohort? Number one, community engagement is key for genomic study, especially in indigenous population, not just doing consultation or research announcements. Like I mentioned previously, in some of the study abroad, you will always hear that the Black participants are hard to reach, or they don't like to participate in study. But we always find something different in African population. I think that there needs to be community engagement. It should not just be a consultation. It should not just be an announcement that we want to do a research. Don't just show up when you need to collect sample. You know, we have some example of where we have engaged the same community for more than 30 years, and we still engage them and we support them. So you see that we don't just show up there to collect sample from them and to do our analysis. So what we can learn from this community, from this cohort, is that community engagement is particularly important for indigenous population. Again, what can we learn? What can we learn from the Uganda cohort? I think that it is not ethically and morally right to only think about our research participants as data. We must think about them as an integral part of the solution that we aim for. You know, I always like to say this also to my group members and students, that we're not just analyzing data. These are people, these are human beings, these are very, very meaningful. So also as colleagues, we should not, it is not ethically right to only think about our research participants as data or as number. We must think about them as integral parts. They are participating so that there can be a solution. So they must be taught to be an integral part of the solution that we aim for. Okay, so from that Uganda study also, in one study, we found a relatively high prevalence of the risk factor for CVD for a young population. You know, like I mentioned previously, Uganda, average population in Uganda is probably about 16.7%, but we find a high prevalence of risk factor for CVD. And unfortunately, there's also a low level of awareness for NCD, coupled with a very low access to care, is very worrying. Bearing in mind that in African population, because this population is quite representative, we currently, about 40% of Africans live in urban area. So meaning that more people live in a rural community. So our community is well representative of what you see in Africa. We also find a high level of liver function in that population, which needs to be investigated. So what we also saw is that none of those with high cholesterol was aware of their status of high cholesterol. People were not aware because maybe there were no facility for them to check. There's no like regular check, like you see in some developing countries. 80% of those with hypertension were not aware that they had hypertension. Eventually, what we did was to treat some of them because we have a clinic facility there to provide that kind of support to them. 57% of people that have higher HbA1c were not aware of their diabetes status. And also the same thing also with people with abnormal liver function. So this kind of study make the awareness to make people to be aware of their status and also supports given to them. So what we did was to provide feedback to the community. So if we take people's sample and we have to give them feedback about their high cholesterol, about their diabetes status, about what is going on in their life, whenever it is possible. So in that community, we have a clinic there at the Yamulibua field station, and this provides support for about 80 patients regularly every day, including providing medication for them. So what can we learn from Uganda cohort? Integrate feedback in your studies. So remember that research participants may not have the capacity to understand your complicated journal. You know, we as scientists, we are always very interested in publishing in high impact factor journals like Nature, like Science, but this may not reach our participants. So we must find a way to integrate feedback to the community, provide feedback to them, including give them results of whatever you have tested from their sample. So in our study, we explore multivariable GWAS for discovery. One reason was because our samples are small, and here we published a paper to look at different methods to do multivariate GWAS. Here is also an example of a GWAS of body shape that we derive from PCA analysis using anthropometric traits. This is very important for small sample size like ours. Yes, one paper that's under review where we perform GWAS of a cluster of cardiometabolic risk factor, or which other people might call them metabolic syndrome, which is currently under review. So what can we learn from the Uganda cohort? So where sample size are small and phenotype are correlated, we could explore model that improve power to detect association. That's what we have done with different multivariable GWAS analysis. So we went further to collaborate widely. So currently, we are collaborating widely within the region in Africa and also outside the Africa region. So we are collaborating within East Africa, within a consortium that was formed previously called APCDR. We have a network that we are setting up called Kidney Gen Africa. We are collaborating on the CKD Africa, collaborating on the Cardinal project funded by NIH. So globally, we are also collaborating on the MAGIC consortium, on IHCC, currently also on the GBMI, the Global Biobank Metabolics Initiative, on the PRIME consortium and CKD consortium. So these kind of collaboration are very important, particularly where sample size are small. So what can we learn from the Uganda cohort? So to navigate around the current global biobank correctly, so regional and regional collaboration is as important as global collaboration. And that is why we are collaborating both within the region in Africa and also outside the region globally. So we have now extended our collaboration, including data from MVP that has about 57,000 African American and also 7,000 Black participants in UK Biobank. So recently, we assessed the prediction of political risk score using data set from multi-ancestry meta-analysis, which has GWAS meta-analysis from 1.4 million people from different ancestry groups, GWAS meta-analysis from 6,000 people from African American, and as well as the European GWAS meta-analysis. So we are trying to derive PRS from this different population and test them on the South African cohort, which is a regional collaboration, and validate that from different African populations. So what we found is that the PRS that derive from the African ancestry individual perform much more better in African population, including from the target population and as well as the validation population. So we went further to split the population in Africa into different countries and assess the multi-ancestry PRS, which has currently been the standard against the African American PRS for different populations. And we saw that the African American PRS predicts better in our population than the multi-ancestry PRS. So this paper, if we need more information, is now published in Diabetes Care. Insight can be gotten from here. So we apply the same model to assess the transferability of genetic risk score for lipid trait in different African populations. And that paper currently published in Nature Medicine. So just like previously, we did the same, try to assess the PRS from African, from the multi-ancestry population, and also from the European population. And again, we find the same pattern that the African ancestry PRS is producing better for different lipid traits. And so we were excited about that study and tested the same PRS that was producing better in South African population in Uganda. And we saw a very poor, very low prediction in Uganda. So again, this is showing that the population in Africa are different, and different PRS may not transfer well into different African populations. So collectively, our PRS work show a need to improve representation of African population in genomic study. So we must think about the inclusion of African population across deriving one PRS across 3,100 ethnic groups is not going to work. So this shows that we may need different PRS for different regions of African population. So what I've been talking about in the last few minutes has been the Uganda Genome Resource. So briefly, I'll just talk about the Nigerian 100,000 genome projects. So again, in order to improve representation of African genomic study, So we are now creating what is called the Nigerian 100K Genome Project. And this is a very exciting project, a private-public partnership. This is now in partnership with a company called 54Gene in Nigeria. And we have some leaders in Nigeria leading the government organization like Professor Batun Salako and you come in Nashiru, myself and a colleague at the university in Lagos. So African scientists must stop waiting for government money that will never come. And it's time to think about an alternative, which is an example is what I gave here of a private-public partnership. So here is just showing an example of the 5-point agenda that we have for the NCDGS. So very important here is that we're collecting sample, 100,000 sample. We are aiming to create an array, a design study for genomic study. And very interesting for me is how we can empower African genomic scientists to take a lead in different studies. And so we published this agenda for NCDGS recently in Nature Genetics. And if everybody is interested, you can have a look at this paper that was published in May this year. So the NCDGS has two projects. One of them is a community project, which is called the Nigerian Cardiometabolic Genome Study, which I co-direct. And the other project is a hospital-based project. So in brief, the NIGGEM project is basically trying to estimate the prevalence of a range of cardiometabolic disease and traits in Nigeria, and also to create a unique framework for building large-scale cross-sectional study in African population by examining a range of health indices. So this study is, here is a map of Nigeria, and here is showing different states in Nigeria where the study is currently happening. So very important for me to mention here is this study also following the model in Uganda, and here taking a step further, looking at several levels of community engagement. So we are doing engagement at this high-level advocacy, speaking with the State Ministry of Health in every state that the studies, the sample are being collected, even the local governments doing community advocacy, speaking with the World Health Committee, and also as the community leader and stakeholder, before going to the household, doing some sensitization, speak with the head of the household, and so on and so forth. So what I'd like to mention here is that genomic study is much more beyond consultation or announcements. It must be an engagement of the community. And here saying that trying to get an informed consent is a process, not just a form signing by a participant. So the participant must fully understand what is going on and give a true informed consent. So what can we learn from the Nigerian Biobank is that the government and charity money shouldn't be the only way to fund genomic study. We must seek an alternative. An example of alternative is a private-public partnership. Again, we can also learn that it is critical to facilitate all levels of community engagement to obtain insights that will lead to ethical research. In case of the Nigerian project, the community engagement is not really one community, but it starts from the state government, from the local government, going to the world, and we must engage all of them. So lastly, to improve representation, we're now working on this Malawi Genome Resource. And like I said, this is a very interesting project that has already have 20,000 people that have been recruited in a rural community and urban community in Malawi. There's been a range of NCD that have been collected already. So what we are doing currently is that we are genotyping about 7,000 of them on East Africa array, and we plan to expand this. So most exciting for me is that this project will be done all in Africa. So the study design has been done in Africa, sample recruitment in Africa, biobanking in Africa, biomarker testing in Africa, DNA extraction in Africa. So now we're going to be genotyping about 7,000, which is a large data set for African population. And this will be done, is being done currently in African population. Most of the studies that you are aware of have been genotyped on East Africa array, maybe somewhere else in the world. But we are taking a step to do this in Africa. Genetic analysis will be done in Africa. This will be African-led for publication. This very exciting project that is upcoming. So what can we learn is that it can be done in Africa. It can be done in continental Africa at all stages. So in conclusion, Africa has the greatest genetic diversity, like you know, and as such, African genomic data and resources are key for genomic medicine. So unfortunately, genomic research has a whiteness bias, which we must think about a way to fix it. I've shown you from relatively small size African General Resource that there's a great value for novel gene association discovery and as well as genetic risk prediction from African population. So from the existing and new biobank from continental Africa, we could help to fix the gap that's currently happening in African population. So I'd like to stop by acknowledging members of my group who are doing an amazing job, including our partner. I also like to thank our funder, Kudu Welcome, who is currently funding me, my organization, Emerald UVRI and LSH. Thank you very much for listening.
Segun, thank you so much for taking us through all these various initiatives. And I love that you kept on coming back to what are the lessons that we've learned. But I think really impressive is the work that you're doing and that you're leading, you know, on the African continent. So, you know, I think the future is bright with all these activities. And I think, you know, linking this to the International 100,000 Cohorts Consortium is also a huge plus. So I just want to highlight a few things that you mentioned, and we already have a couple of questions, but I really thought that the emphasis on community engagement is so key to building cohorts and being able to go back to them. So, you know, the initiatives and the effort that you're spending there is really critical. But, you know, as you say, you have to think carefully about feedback of results. And I know in H3 Africa and the projects that we do, we think about that a lot. And one of the issues is also, what about feedback of genetic results at individual or aggregate levels? So maybe you can address that a little bit later. But, you know, you highlighted again the low access to healthcare, the poor healthcare infrastructure, which really impacts awareness of disease. So, you know, a much tougher problem for us in Africa compared to more developed parts of the world. And then your lessons learned that we need to think of innovative models that are powered with smaller sample sizes, because we're not going to suddenly, you know, have the sample sizes that they have in these international cohorts. The fact that we have to participate and leverage international cohorts, which I think is really important. And that, you know, we see this tremendous regional variation across Africa. And we have to think very carefully about transferability, because your work has illustrated very well how, you know, what we do in East Central Africa doesn't translate to South Africa. And I think, you know, again, that means that we can't talk about Africa or African genomes. We really have to think more carefully. And then finally, you know, thinking about these public-private partnerships. I hear a bit of an echo. I don't know if that's a problem on my side. Are you guys also hearing the echo? Maybe not. But Segun, just to say thank you again. And I'm going to go to the questions in the sidebar, and then I'd like to ask you some as well. So thank you for finishing in good time so that we have some time for discussion. So the first question came from Jason Torres, who really is asking again about that difference in heritability of traits like height and LDL in the Uganda versus the European populations. If you just want to explain again why you think that is so. I know you did this very well in your paper. Over to you.
Yeah, I think that there could be many reasons for that. One of them could be diet. It could be anything. The population I've described today is mainly rural community. You see very different heritability in terms of height for Uganda compared to rural population. You see lower heritability for height in Uganda. You see a higher one in European population, and you see the contrary to that when it comes to LDL. So I think there are many factors that could be contributed to that. And one of them could be the people's lifestyle, including their diet.
Yeah, I think what is so interesting is that, it's opposite. So, I always think that, maybe people in Africa do not reach their genetic potential for height because of food insecurity and poverty. And that might be why it has lower heritability. But on the other hand, with LDL, people in European countries often are on statins. So you're not actually looking at their real LDL levels, whereas in Africa, these treatment naive populations maybe show higher heritability because you're actually measuring their innate LDL, which I think is really fascinating. Great. So Ron Ling is saying, great presentation. And she just wants to understand, in terms of African infrastructure, if you identify somebody who's hypertensive or has a high glucose level, what do you do to make sure that they get medical attention?
Okay. I'm not a medical doctor, but I think this also depends on where they live. So I've described today a rural community where healthcare system is largely lacking, and so many people may have hypertension and not even be aware that they have hypertension, or they have diabetes and not aware about what they have, whether they have diabetes. So what I've described today is that in our study, where people have hypertension or diabetes, so we provide ancestry support for them because we have a clinic that can provide that kind of support where it's possible. So I think that that's a rural community. There's so many urban communities that are well equipped, that people on their own, they are well exposed about what is going on in their life. And there are so many general hospitals where people normally go for medical checkup to see what is going on in their life. But what I want to emphasize is that currently there are still more people living in rural community that access to healthcare system may be very, very limited. And that is why I emphasize the need for us to provide our feedback when studies is being done in that community.
Thanks very much, Segun. You're right that we always sort of balancing, your role as a researcher and how far that reaches towards, providing access or referrals into a clinical setting. And that is usually puzzling, but we have to think about that because of the poor resources and infrastructure. So I have a question for you. So you're creating now an enormous amount of genomic data. When we think about the 100,000 K study in Nigeria and the others, where will the data be stored and curated? And do you think that we have enough capacity for the analysis of the data in terms of genetic epidemiologists, biostatisticians, and bioinformaticists on Africa to actually do justice to the data?
Yeah, I think a lot has changed in the last 10 years. And thanks to H2 Africa, there's been an enormous capacity building that's been built in the last 10 years. You see a lot of PhD students have been trained to analyze data. A lot of postdocs have been trained to analyze data. And I'm always very proud to say that I was one of the people that benefited from the H2 Africa at the beginning, trained for my postdoc. And I know that there are so many other people also that had the capacity to analyze this kind of data. So I think currently, I won't say that we are currently there where we have all the facility, resources and expertise and capacity to analyze data, but a lot has been done in the last 10 years. So in that project I described, the 100K projects, one of the strategies is that, so we are funding people that are PhD. So last year, 4 people were funded to do their PhD. And the idea is that they also analyze this data. So they are trained to analyze the data. And I think with that effort and many other efforts within the country, we'll begin to have more people that will be in capacity to do that. So that data set currently is being generated. mostly analyzed in Lagos, and opportunity will be given to some of the people that have been funded on this project.
Thanks, Segun. Yeah, I think we have quite a way to go in terms of, making sure that our capacity is sustainable and that we keep them on the African continent, because I know that we have a huge exodus of trained bioinformaticists from Africa. So just in the chat, Chisum is, let me just find it again, saying, no, I've just lost it. Just congratulating you on a fantastic talk. So thank you very much. And then somebody else, Sheng Luo, was asking, are any of the study data available to other researchers?
Yes, that's why I say yes. So I took my time to talk a lot about the Uganda General Resource. And before that paper was published in 2019, so the individual data set, it was made available on EGA, and anyone could go there and apply for that and get access to that. Apart from that, so when we perform the GWAS in 34 cardiometabolic traits, so all the somatic statistics were also made available on GWAS catalog, and anyone could just go there and just download them. So we made all the data available.
That's wonderful. Thank you, Segun. And I'm sure very open to collaboration. It's one thing sharing and people doing their own thing, but it's so important often to give context. And therefore, I'm sure, you're very open to collaborating and working together rather than just having people use the data. So there's a place for both, for sure. So I also wanted to ask you a question about the emphasis on public-private partnerships and how you think, so 54Gene is a great example. I'm not quite sure what their funding model is, but how do you think we encourage that more in Africa to build cohorts?
Yeah, I think that's one sure way, because for many years now, decade now, we have waited for the governments in Nigeria and in Africa to fund research, and they're not forthcoming. I don't think we should wait any longer. So if you look at the high-income country like the UK Biobank, for example, you see that big components of UK Biobank is being funded by private organizations. There's whole-genome sequencing being done by the General, exome sequencing being done by different private organizations. The same model for all of us projects in the US for other national biobank initiatives. So I don't see any need, the reason why African also will not explore that kind of initiative.
Very good points. Thank you, Segun. I can see that we're drawing to a close now. And I just want to draw people's attention to the chat box where Chisum has also indicated that Segun will be moderating a panel in the virtual IHCC summit that will be titled The Role of Regional Structures in Supporting Cohort Science in Low Middle Income Countries. So I hope people will register and be sure to attend that session. I think it's going to be very interesting. And lots of congratulations in the chat, Segun, not only for the work that you did, but for the clarity of the talk that you gave, and from me for emphasizing the important things like community engagement, data sharing, feedback, all those things that we truly value if we want to make sure that Africans will not be left behind in this genomic revolution. Now I'll just hand over to Latifa, who may have a few announcements to make. And thank you again, Segun. Great appreciation for your time today.
Yeah.
Thank you.
Thank you, Dr. Michelle Ramsey and Dr. Segun Fatumo for this great presentation and moderation. And audience, thank you for your participation, and please mark your calendar for the next educational webinar series that will take place December 15 at 9 a.m. EST and 2 p.m. UTC time. Looking forward to seeing you again next month. And have a great day and great afternoon.